Okay, so we're going to talk a little bit about what we're planning on doing for the lab practical. This is a little review. Man, this microphone's really weird every time I get it. Hold on. No? All right. Still loud. Wow, this doesn't change anything. Great. I love when that happens. Okay. Well, I guess we'll just start and it'll just be weirdly loud. So a couple things real quick. Uh, we're going to talk about basically how to succeed and not fail, which is the most important part of the lab practical. Quick reminder, the lab practical is a very large chunk of your overall grade. So make sure you guys are aware of that. So it's important. How do I not fail? Well, so there's some easy ones. First one you could do is review your previous lab lecture PowerPoints, very important. You could also panic moments before the lab practical. That's always a classic. Write down C for every answer, or just don't put your name on the paper, so it delays the inevitable. That's my personal favorite, just because it takes longest time. No, don't do that. But what you should do instead is review the lab practical, lab lecture PowerPoints and the lab lectures. That's actually a really handy tip. Knowing the steps in each test that we do is a really important one, which we'll talk more about in a second. Knowing the reagents is maybe not the best word I could use for it, but knowing the components of the media that we use and why we use each one. So a good example of this would be like, hey, why do we put mannitol in an MSA plate? We're looking for mannitol fermentation. Why do we put phenol red in an MSA plate? We're looking for a change in pH. Those would be examples. And of course, you should know what the results look like. So for an MSA plate, you should know what a positive MSA plate looks like, what a negative MSA plate looks like. You know what growth looks like and what no growth looks like. Those are examples of things that you should know. So we're going to talk about those in just a second here. The way we're going to start this, for you guys, it's going to be Tuesday. We're going to do a skills check. We're going to have 2 skills check. And what this is one of each, it's not going to be a mixed culture for you guys. It's just going to be a straight culture. And you just have to streak for isolation. You have to get one isolated colony. That means a colony by itself, no other bacteria next to it. So just isolated, not touching anything else. We're going to grade you on correct streak pattern. So the streak pattern should be the one listed in the book. And it should also be incubated properly with a correct label on the back of the plates. And of course, make sure that you guys please tape up the plates in the back so they're not individually tape up the plates, because we're gonna have so many plates in there, and if something gets knocked over, we don't want to knock over 100 plates and have them all open up. So make sure you guys tape them shut a little bit, just a little bit of tape on each side. But of course, make sure that you properly label them. Name, section, what it is, normal naming stuff. Partly this is also to make it really easy for your TAs to grade it, because if they have a hard time reading what it is, they're just gonna give you a 0. So don't make them guess. Now the other one, which is gonna take a little bit more time, is pipetting of volume. So we're gonna give you guys a little microtube. We're gonna say, hey, give us this much liquid. It could be 100 microliters, it could be 560 microliters, it could be 13 microliters. We're gonna ask you to pipette it into the microtube. So we'll give you guys a large container with colored liquid in it so you guys can pipette it in easily. You're gonna have to choose the appropriate pipette and set it to the appropriate volume. You'll check it with your TA, and then they'll let you go ahead and get a tip on and transfer it. And the most important part of this is making sure that you guys transfer it in a sterile fashion. So you open up the lid for the box, take out a tip, close the lid right away, open up the stock solution, suck it up appropriately without accidentally like touching the side of the tip or touching it to the outside of anything in the world, so it stays sterile. Popping open your tube that you're going into and pipetting the liquid in the correct volume. And if the volume matches up to what we have, then you guys will be good to go for that. And if not, you lose some points. So this is just going to be added on top of your lab practical grade, so it'll just kind of be added to the top of it. This will be a skills check, essentially. Just making sure that you guys have some practical skills ready. Yes?
Does this include serial dilation?
No, we're just going to have you pipe out one volume. So this is just doing one volume. So we're gonna test your abilities. We're gonna see how you guys are going, and then we'll talk more about what the actual format of the quiz is gonna look like. So I might show you guys, I might give you guys a slide and say, hey, look at the microscope. What is this? So what kind of stain is this, guys? Acid fast, very good. What's the primary stain? Carbonyl fusion, very good. Okay, so most of you guys didn't know that off the top of your head. That's an important one to know. That's a component of this that you should know, and you should know which order it comes in. Well, I'll follow this up with, does anybody know what the secondary stain for this guy is? It's not crystal violet. It's not saffronin. It's a methylene blue, yes. So the bacteria, so the carbonyl fusion's kind of red and methylene's blue is kind of blue, as you expect it to be. Why do we have to use steam for this particular stain? Open the pores, penetrate the bacteria. Okay, these are all good ones. So yes, it does resist staining. What is the chemical on the bacteria that resists the staining? Mycolic acid, good. This side of the glove is killing it. Okay, you guys talk more, mostly because I'm like really close to you guys. And I won't have to go what every 5 seconds. Okay, now last one, really important one. Oh, well, actually, there's another one. I'm gonna pose this to you guys, see if you guys actually paid attention. So this would be harder than what I give you guys, but it's still a good question. Why does mycolic acid resist staining? It is waxy. Why do waxy things resist staining? I heard them say it, hydrophobic. Yeah, it is. It's hydrophobic. So it's more, a better word for it is non-polar. So dyes are almost exclusively polar, and so they're going to resist staining with mycolic acid because it's non-polar. Polar and non-polar don't want to mix together. They want to stay apart. But carbonyl fusion is actually a phenol-based dye, so it's got a slightly non-polar portion. And because we're using heat, it loosens up that mycolic acid layer so it's a little bit more pliable, so we can allow the dyes to get in. And once they're in, they get trapped. So there you go. All right, great. What is the result for the bacteria that are being pointed to in this arrow? What was that? Somebody over here have an answer? What was it? Acid fast negative. Very good. Did you look at the answer sheet? Yeah. Okay. But it's or non-acid fast. Both are fine. By the way, I'm not going to be too picky if it's like roughly the same concept. Non-acid fast, acid fast negative. This is fine. We're not going to be picky about that. If the spelling is off, well, hey, look, I didn't get into biology to spell things correctly. I use computers for that. So if you guys are mostly close with the spelling of something, I'll be fine with that. Okay? Don't sweat that too much. Okay. Next one up. What kind of plate is it? Blood auger. Good. What kind of organismal growth does it support? Fastidious. Very good. Good job, right side. All right. Now, what is the result of this particular plate? All right. Raise your hand if you think it's alpha. Raise your hand if you think it's beta. Raise your hand if you think it's gamma. Gammas have it. It is a gamma, or non-hemolytic. So the reason why it's non-hemolytic, so you can see the colonies right here, but the blood around each of these colonies is intact. It's not see-through. It doesn't have any discoloration to it. The discoloration or the see-throughing, the translucentness of the media is caused by the hemolytic activity where they break apart the red blood cells, and now they can see through the media really easily. That's why you can't see through this media right now. It's completely opaque. It's just red all the way through, and you can't see anything on the other side. It's because it's a non-hemolytic bacteria. Okay. What makes the plate opaque? Blood. Okay, can you be more specific? Huh? Red blood cells, yes. Red blood cells make it opaque. Now, the reason why it's not just blood, right? And blood's like a mixture of things, right? Red blood cells, white blood cells, platelets, nutrients, all this stuff. Blood, it's in of itself, just the liquidy portion. You just spin it down, it's see-through, it's clear. So that it's not, yeah, keep that in mind. Okay, so it's red blood cells that make it opaque. Great. All right, so here are the things to study. I know it looks like a lot. You know, some of these you guys don't have to worry about. So the ones that say for Bio 4102, just ignore those. You don't need them. They are for Bio 4102 only. So they're the more advanced questions, so they get more difficult questions and more stuff. You guys have to worry about lab safety. That's the one that everybody tends to forget. They're like, you can't remember that far back in the semester, but it's actually important stuff. You know, hey, what does proper protective equipment look like? What does, where do we keep the eye wash station? Where's the emergency shower? What are you supposed to do with your gloves after you guys are done with them? Things like that. How often should you drink bacterial cultures? Never. Okay, basic microscopy. So this is going to be things like knowing all the parts of the microscope, knowing how to focus a microscope, knowing the definitions of certain things like working distance, resolving power, diffraction, so on and so forth. Those would be examples. You should also know about like the white blood cells, like, because that was like our very first one, is like looking at it being like, oh yeah, these are white blood cells and red blood cells, and just kind of being able to vaguely interpret them as such. You don't have to memorize like what a lymphocyte is or anything, but you should kind of have a base idea of like what certain things are. Pipetting. Obviously we're gonna have a skills check, but you should also know like, hey, what's the appropriate volume? What are the parts of the microscope? What's the effective range? What's the maximum range? These kinds of things. Those are all important parts of pipetting. Yes? Oh, geez. Actually can't remember what I put there. I'm zooming in. Let's find out what I wrote there. This is actually a really long time ago that I wrote that. Apparently I really like explanation. Thanks, Jay. Yep. I completely forgot that I wrote that. Okay, great. Good eye.
Also on the viable plate, I'm guessing the more replacement what numbers will be given?
All the numbers you'll need to solve the answer. I'll give you guys everything you need to get the answer right. But we'll talk about that in a second. So Gram staining. Know the components of Gram staining. What are the chemicals that we use? What order do we use them for? And how to interpret the results. Hey, do you know what a gram-positive foci looks like in a Staplo arrangement? You should. Do you know what a gram-negative looks like in a random arrangement? You definitely should, and should be able to determine those and look at them. You should also know, like, what is gram variability, because that's a big issue, or how to heat fix a slide. Those are all examples. Endospore and acid-fast staining, so you guys should know both of those techniques. You know, be able to look at it and be like, okay, I know what the chemicals are that are used in it, the order, the time, and how to interpret those results. You know, if you saw a bunch of green little dots on a slide with pink rectangle-looking things, you should be like, that's got to be an endospore stain because that's what those look like. They're the only things with green things in them, and there you go. And then you should be able to identify the parts in those. So, free spore or an endospore or whatever. Okay. Negative staining, you guys don't have to do. Transformation, you might get a plate. You might just get a picture that you have to answer some questions about the parts on the picture, but you should know the general flow through of what we're doing for transformation. What does it look like? How long do we do stuff for? You know, how long do we do the heat fixing step for? What temperature do we do the, sorry, not heat fixing. What temperature do we do the heat shock treatment at? And what temperature do we incubate it at? Things like that. What kind of antibiotic do we use on that? Culture transfer techniques and isolation, that's not gonna be too much. We're gonna give you like a plate and some cultural characteristics you might have to analyze, or maybe a broth, same thing. So it's not gonna be tons of stuff on that one because it's kind of vague, but be roughly familiar with different types of culture characteristics. If I do ask you guys how to do, like what the cultural characteristics are, I'll probably give you guys a little chart for like answer it because I don't expect you guys to memorize a chart of like 40 things. So there you go. Okay. What do we got? Viable plate count. So I might give you guys a scenario where I've said, hey, this is how many dilutions we've done, and this is how many plates we got, and this is the number of plates with that are in the countable range. Figure out which ones, you know, what the CFU per mil is. Figure out what the final dilution is. That might be something that I'll ask you guys to do, or how to interpret those results. Similarly, I'll give you like an oxygen, the oxygen tubes with DHI. You should know what the DHI is. How do we do the inoculations? Because it's a special type of media. It's liquid that we give it to you, but it solidifies. How to interpret the results for things that are going in like a strict anaerobe fashion, or an aerobic fashion, or a facultative fashion. Water testing you guys don't have to do. Selective and differential media. Now, this one's a little bit trickier. Because there's so many things on here, it is important that you guys really put some effort into reading through this. Because it's new, but it's also got a lot of steps in it, make sure you guys really spend time with it. You should know things like the names of plates. So you should be able to look at a plate and go, oh yeah, that's what this plate is. You should know if it's selective or differential or both. You should know what it selects for, differentiates, or both. You should know if it's got any special chemicals added to it. You know, like, does it have mannitol in it? Does it have phenylethyl alcohol in it? Does it have blood in it? Things like that. There you go. So that one's that. Nitrogen cycle, again, not you guys. You guys don't have to worry. The last one is the bonus step, which will have a section on the cool science thing of the week. It'll be about 5 or 6 questions. Each of those questions are worth 1 point apiece. Your entire exam, I think, is only like 75 points. So it's a pretty big chunk of your exam. So if you guys like want to have a pretty big impact on your overall grade, those bonus points can help out a lot. They can turn a B into an A pretty easily. But they tend to be a little bit harder in nature just because they're bonus points, so just keep that in mind. You could skip them if you don't want to do them at all, that's fine too. There's no points off if you get them wrong. It doesn't affect your grade. Yeah, so there you go. So that's the subjects that you should cover. The format, what is it going to look like? We're going to start out with a short battle royale where you fight your classmates to determine who gets to win and get the overall points for the entire lap. Or, that would be a bad idea, we'll just instead do it where you guys go through each station. We'll have them set up at your tables. There'll be a total of 24 stations, and then a bonus one that's the 25th. You can skip that one if you don't want to do that. You just sit there and chill for 2 minutes. After a total of 25, or after everybody's gone through all the stations, we'll give you guys another chance to go through and go back and look at a station for however long we've got left in the lab. Now, here's the thing. Don't just like line up in front of, or like cluster around one station. So if everybody's like, boy, I'd really like to look at station 7 again, and there's like already somebody there, don't like crowd around them. And also when you guys are looking at them, make sure you guys can like cover up your test sheets so you don't have your answers exposed to everyone. You know, try to form a nice little queue so that you're not like looking over each other's shoulders at what the answer is for that one. The TAs will kind of go around and help and keep an eye on that. But yeah, that's pretty much it. And then after you guys are done with that, you'll turn it in. These are set up to be free answer. So it's just gonna be a fill in the blank. You gotta come up with the answer on your own. There's no multiple choice, there's nothing like that. It's just free answer. Like I said, I'm not gonna be too hard on spelling. And if you get it roughly close to the correct answer, like, you know, if you said acid fast negative instead of non-acid fast, those are the same thing. I'm not gonna be pedantic about it. Yes? Well, they should be getting close to that. Probably not the last 2, because it was just going to take a while to grade, but realistically, I don't take any questions from your lab reports, so those won't help you at all. The things that I talk about, though, like I just talked about, or that are on the review sheet that I'm going to show you guys in just a second, are great things to go look at. In fact, one of them, especially for the selective and differential, there's like a little chart in there that you had to fill out for your lab report. And assuming you guys fill it out right, and you guys can ask your TAs on Tuesday for you guys. The correct filling out structure for it so that you guys can make sure you have a good one. That's a good study guide, a little chart to keep in mind. Now, are there any questions? Yes. No sentences. I don't want to read sentences. I try to keep it at like 1 to 2 words. There might be like a third, you know, acid fast stain is, 3 words. And that's about it. That's as far as I want you guys to go. Okay. And don't kind of be like, and if it's like, hey, what color is this? Don't go like reddish, bluish, green, because I'll just mark it wrong. Don't. Just make it easy. Just pick a color. Be bold. Choose wisely. Okay. Now, aside from this, one last thingy. Or that. Okay. There should be on Canvas a lab practical review document. And it should look something like this guy. And it's kind of got a bunch of things to look through and fill in the blank. You guys can test your knowledge. And if you guys have questions about it, on Tuesday, you guys can ask your TAs. Don't just ask your TAs to do the entire thing with you. Go and do it yourselves. And if you're like, I can't figure out this blank at all, then you can go ask them that question. There is one section in here, obviously you guys don't have to worry about, it's like antibiotic control. And some of these also have like the nitrogen ones in it that we didn't do, or the water lab. Just ignore those because we didn't do them, but keep that in mind. Okay? Does anybody have any questions on this so far? All right. Word of encouragement, or not really encouragement, more to scare you. So typically people, when we do the first lab practical, they tend to not do as well on it because they're not expecting it to be difficult. It is pretty difficult because we're asking guys to be able to regurgitate anything that we had talked about in the first half of the semester and being able to use those skills. People tend to do much better on the final, so don't be in the group of people that has to do really well in the final because they didn't do well in the midterm. Really put some effort in, make sure you study, make study groups, test yourselves out, make little Quizlet things. I know you guys like to do that. And if you guys have questions, rely on your TAs, especially on that review day, because as soon as they're done with the skill checks, they're going to go over the review. So make sure you guys spend some good quality time asking them questions if you have. Okay? All right, if you guys have any questions, come up and see me or email me, and I will see you guys next week.